Okay, welcome back, students, for the next session. Our next session would be conducted by Dr. Saptrishi Purkaisa, Director, Health Informatics, Associate Professor, Data Science, Health Informatics, IUPUI. Saptrishi Purkaisa, sir, initiates innovative research that focuses on data in the field of healthcare. He has worked on and helped to implement projects around the world involving national health information systems, CAD applications, mHealth, educational technology for dyslexics, and much more. On behalf of the World Health Organization, Dr. Purkaista has served as a consultant to ministries of health in Southeast Asia region for implementation of health information systems. Countries that were part of this project included Bangladesh, Nepal, Bhutan, and North Korea. We are honored to have you here with us today, Dr. Purkaista. I now request Dr. Saptrishi Purkaista to start the session.
Hello, everyone. I hope you're able to hear me and see the screen.
Yes, we are.
Okay. So I can see a few people, and the title of my talk today is about novel deep learning approaches, primarily for human AI collaboration in radiology. So I work in the domain of radiology, and the talk that I'm going to give you today is primarily about how human AI collaboration can be enabled through some novel methods. And this is a multi-site research project that we're working on. So it includes collaboration from Emory University. But before I even begin, my disclosure is I have no actual potential conflicts of interest in relation to the presentation and the products that we're going to talk about today. So a quick introduction about me, like Dr. Narad said, is that I'm Associate Professor of Health Informatics and Data Science at Indiana University, Purdue University in Indianapolis, and my research has been funded, and I'm thankful for all of these funding to enable the research that I'm going to talk about today. I work in the intersection of public health, biomedical informatics, and sensor technologies. So I work in global health, particularly implementation of different health information systems. I also work in open source medical record systems and telemedicine systems. And I also work with mobile apps and smartwatches and identifying user behavior using these devices. So primarily, I think the conundrum today, whenever you look at how technology is being used, particularly how AI and humans collaborate, I think the question is usually between how one will dominate over the other. And there's very little conversation between how this collaboration can enable more productivity and better learning for both humans and machines. So this conundrum of human technology frontier is, will humans be augmented by technology? So currently what you see is a GPS system or a mapping system that tells or guides a human driver and finds the best path to go from one location to the other. So this is, you can think of humans are being augmented by technology. And on the right side, you can see how a self-driving car where humans really only have to mention that I want to go from here to here, and the car takes the person. The human doesn't have any or very little sort of action to do. And that is where you can think of technology is being augmented by humans. So instead of thinking of these two as separate entities, what we have proposed and what we found is that instead of thinking of these as a conundrum, we probably have to think of them as one entity or an assemblage. So the human machine is just one thing, and the capabilities of this one thing is different from looking at them separately. So So if you see this idea of assemblage, it comes from uh, some of the very earlier work by Pablo Picasso. So here you can see this is an assemblage art, and it comes from the idea of art that you can mix very different, unique things and produce something totally new. So a lot of social scientists have come up with this idea that when you have a group of objects of different or similar types, found in close association with one another, it's called an assemblage. So Deleuze and Guattari first created this idea of an assemblage, and then DeLanda and a few others in this STS field, science and technology studies, sort of um, expanded this idea, which is core to our research designs that use actor network theory. I'll give you a couple of examples that are fairly common when people talk about assemblage. So a gunman is different from both man and a gun in the sense that a gunman is able to shoot someone, whereas the man and the gun alone cannot do this. So the properties of this assemblage are very different. Another example is water. The separate components of water have different capabilities. So hydrogen and oxygen are both combustible and support fire. On the other hand, water extinguishes fire with very different capabilities. So the idea to think of assemblage is that it's greater than the sum of the parts, and um, that the capabilities of the assemblage are very different from the individual elements that participate in an assemblage. So with this idea, when we think of how the radiologist and AI form an assemblage, the radiologist AI combination has very different capabilities from what the radiologist alone and what the AI alone has. And that's what we are investigating through this research project and what I'm going to talk about. Um, so actors don't remain the same when they get into an assemblage. This mutual effect of impacting each other is called as assemblage learning. This is a concept we came up with, but others have called it with many different names. However, this idea is that humans learn from machines and change themselves, and machines learn from humans and change themselves. And to be able to enable this fit or this assemblage, there have to be some methodological changes in the way we train our models. And similarly, humans have to be trained to be able to adapt to how a machine learning model or an AI model works. So this is sort of the high level architecture of how these works. Assemblage learning. So on the left, what you see is how traditionally an unmediated radiology practice works. The radiologist looks at an X-ray that's been captured, makes an interpretation. After the interpretation is made, usually another radiologist would also make an interpretation. Depending on the difference between the interpretations, one or the other radiologist sort of changes their mental model, and they learn from peer feedback and practice. So they keep doing this hundreds and thousands of times over a year or 4 years in their medical study, and then finally build a mental model. When they are shown other images, they're able to interpret what things are there in the image. Traditionally, on training the AI model side, people are given labeled data. Sorry, the machine learning model is given labeled data from humans. So there are some labels from in the images, and then it learns that this is the feature representation in the image, and that image is then shown to the machine. A new image is shown to the machine learning model. It tries to find the same feature representations, and if it's able to find that feature representation, then either it does classification, which tells what kind of pathology or diseases are there in the image or objects are there in the image, or sometimes there is a segmentation model which highlights the region of interest or area where it's able to find the feature, right? But then when new data set comes or there is new labels, then the machine learning model does not understand the feature representation of it. And this is usually a static model that does not really learn on its own. So some new methods have come up where if a radiologist makes an interpretation, the ML algorithm that already is trained is able to show its understanding or its output, either classification or segmentation, out to the radiologist. The radiologist looks at that interpretation in respect to the mental model that they have. They adjust the bounding boxes of the region of interest, if it's a segmentation problem, or highlights that this is a new label that the AI model did not have, and then it adds that to the label. Based on this interpretation is then going back to the machine learning model. The machine learning model retrains itself with a new space, including some of the weights that it already had and some changes to those weights. And in this process, the machine learning model is able to fit itself to the radiologist. And because the AI model has shown some output, each time it fits better and creates a assemblage with the radiologist. So the radiologist and its own specific AI becomes partners in future work that is done when future images are shown. So there are some specific methods we should think of. And why do we need to do this? Because there are some parts that radiologists are good at, the humans are good at, and there are some parts which machine learning models or AI are good at, right? I'll show you some examples of where we've seen AI models fail very badly. So here you see as an example. So this is uh, where there is some kind of fluid that gets into the lungs, um, uh, and then this have to be out. So a lot of times, some of the most popular models out there on chest X-rays are able to predict um, this kind of disease, pneumothorax or sometimes embolism, using some conjugate such as chest drain, right? So if you see the accuracy or the AUC for where there is no chest drain, but there is the pathological problem, it has an AUC of 77. On the other hand, when there is a chest drain, which means the water has been taken out or the fluid has been taken out from the lungs, the AUC is 94. So very high accuracy model, but the point of the model working here is not as important as it working here, because the model should be able to help the human in identifying cases and improving outcomes of the patient. But on the other hand, here already the treatment was done, and the risk of harm is lower. So higher accuracy in this is less important, which is why a human in this case would know this is more important. And instead of depending on the AOC score alone, you also need to understand how important or how critical a case is, which very few models are able to do today. Another example I want to highlight is where the disease labels are the same. So the idea that a pathology has to be identified is important. So the same disease may appear in different parts of the body, and very different modalities. So this is a chest X-ray, this is a hand X-ray. We're talking about sarcoidosis, which is lymph enlargement that you can see in a chest X-ray, and that's how you would identify the disease. Or you'd see different kinds of absorption on asymmetrical bones. Like some bones would have higher resorption, others will have less, but the labels are the same. So if you had to train a ML model with this label and show it this and this, it would not be able to identify the labels in both the places. It will learn only one or the other. And this is usually a problem, but if you show this to any human radiologist, they would be able to know that the sarcoidosis in this is very different from the sarcoidosis in this. Then I think a lot of us have seen many of these COVID identification models. They work really well in identifying if there's COVID or not COVID, But when there is models, when there are pathologies which are similar in nature, um, in terms of how they are represented in an X-ray, like normal bacterial pneumonia, viral pneumonia, and COVID-19 pneumonia, because they're all types of pneumonia, the failure or the false positive rates in these are very high. So these are some of the most popular, most accurate models, which have very high false positive rates, as you can see. And false negative rates are also very high. So the idea is that they work really well in identifying um, normal chest X-rays versus COVID-19 X-rays. However, if you ask it to identify between viral and bacterial versus COVID, it does pretty bad. And a human, on the other hand, would do very well in these. So to be able to deal with these, we have come up with a few ideas, and we've tested this and published a few papers on this. One of this is called margin-aware intra-class novelty. The idea is to be able to distinguish between this and this. What does a model need to learn? So between a hand X-ray disease label and a chest X-ray disease label, like what does the machine learning model have to learn? And so what we've done is we take three different data sets. That's what we're using here, and we build encoder decoder based architecture. So this encoder decoder based architecture, we have a stage one, which has an encoder and an encoded feature after taking an image, and then it uses a decoder and produces the reconstruction of the image. Right. The idea of doing this is that if we are able to identify the features, if we are able to identify the features that are very specific um, to a hand X-ray compared to chest X-ray, we'll be able to know that the label can be identified through certain kinds of features versus a label can be identified through a different set of features. And so we create a feature space, and based on those embeddings, we share the embeddings to a new model, and the new model then uses a FCN, a fully connected neural network, and then makes the decision that I need to apply the label in using this feature space and this other feature space, right? So we call this architecture TEND, transformation-based embedded learning, and you identify if something is novel or not novel, right? So these are three different data set: a chest X-ray data set, a skin data set, and I forget what the third one was, um, but yeah, so three different data sets that we use in three very different body habitus but similar labels you could apply to all three of these, and then we do very different kinds of transformations on these images, and then apply some blurs and things like that, and are able to do this better than many other popular architectures. So this kind of architecture, we believe, can be useful where the labels are similar, but they mean very different things. The second kind of architecture, which also is similar to TEND, but what we identify is that you don't necessarily have to use medical images only. So a lot of times, what we wanted to do is to be able to identify noise or similar class things, and any new thing that shows up, like a new label or a new image, the model should not be able to make the classification that it's already learned. So, for example, if you show current machine learning models that have been trained on chest X-ray, an image of a bird or another data set like MNIST or fashion MNIST, it would still try to make an inference. because it does not realize, the machine learning models don't realize that this is a different class of images compared to, say, this one, right? Or for example, this is a different class within the same type of, so this is called intra-class out of distribution data. This is called inter-class out of distribution data, right? So to be able to identify that this is different from this is different from this, or this is different from this is different from this, or this is different from this is different from this, right? So to be able to identify these differences, we have a discriminator where we call it cascade VAE. So this is basically a VAE model, which is able to identify architecture, that is able to identify anomaly or out of distribution images, right? And sort of without getting into too much details, we compare this with many different kinds of out of distribution um, architectures and find that our architecture does fairly well on many of the interclass problems that we, out of distribution problems that we've come across. The last one is something that we are quite proud of, in the sense that the problem that we saw about pneumonia being of different types and normal chest x-rays, bacterial, viral, COVID pneumonia, there are really no good approaches that have shown to work on identifying the differences within similar pathologies, which is why we came up with this approach, which is called few-shot learning approach. So the human then corrects this, corrects the model, and the model learns on those only. So it does not go back to the entire data set and retrain itself, but only certain layers are fine-tuned in the model, so that these differences that humans have identified to correct the model are only learned. In other places, this is also called reinforcement learning through human in the loop, which is too long a name, and we thought a few shot learning with human intervention is good enough. But the idea is the same, is that the entire model does not get trained again, but rather there is a retraining that happens, and retraining only on the highest, most layers with these new labels, or with changes in the feature set for existing labels. So with this, I think we've seen big in negative predictive values, as you can see here. So NPV improves quite a bit. So which means there are fewer mistakes made by the model, where it was making mistakes earlier. So with multiple iterations. And what we are trying to do here, and we have a human study going on. So we've interviewed about 35 radiologists across Emory University in Atlanta, Indiana University, and we've shown these models gotten corrections about these labels. And then we've seen close to double the gains, both in terms of accuracy and in terms of fewer, half the amount of time taken for these cases. And this is across many different kinds of modalities, which means for breast mammograms, for chest x-rays, um, for lesion detection in breast, as well as head CT scans for brain tumors. So we applied this to, so each radiologist spends about 15 to 20 cases each time. Once they do it without an AI, the second time they do it with an AI. That study is still ongoing, but we've already started to see some very interesting results that we're able to do this. And then also, this is another paper which shows where AI is better. So human capabilities need to be augmented by the AI. So this is some example. Some are our studies, some are other FDA approved studies. And the last part that I want to talk about is something about unknown unknowns, right? So there are some things. This is sort of a seminal pathbreaking paper that we published now about 2 years ago, um, and uh, where we were able to show that AI is able to recognize a patient's racial identity just through plain medical images, and humans don't know how the machine learning models are able to do this. So a lot of press covered this paper, and we still don't know how the AI model is able to do this. For example, if we added a high pass filter to a chest X-ray um, and produced something like this noise that you see here, it is still able to identify with 90% plus accuracy if this chest X-ray has come from a white person or a black or African American person or an Asian person from this noise, and no human sort of human radiologist has done this more than 50 accuracy, which is like a guess. So, so it's odd that machine learning models or AI models, fairly simple AI models, are able to do this. And he wanted to investigate if this is something like an overfitting problem, right? So because we've trained it so well on certain kinds of images, does it work? So what we did was we also did external validation. So we took one data set, trained this model, and then asked it to predict on another data set, and still it was able to do it with 0.97, 0.98 sort of accuracy. If it was just chest X-rays, then we thought, okay, what can it do beyond chest X-rays? And we took chest CTs. We took digital hand atlas, which is a data set about hand X-rays, and there also you can see it's able to do this with fairly high accuracy. So I think broadly there are parts where humans don't do as good as machines, or machines do much better than humans, and so that's why we need to investigate these unknown problems. I think I'm over time, so I'll skip a few slides and then get back to if anyone has any questions.
Thank you so much, Dr. Purkaister, for your lecture. Participants are encouraged to ask questions. Okay, so I have a question for you. Uh, how do you deal with the variations in anatomical structures and the scarcity of labeled anomaly data? I mean, when you don't have these things, then how do you work it around? Because this is something we face a lot in the Indian context when the data is given to us as and data scientists. There's a lot of problems like these. So how do you deal with them?
Yeah, thank you for that question. So the Cascade VAE paper that we recently published, there what we're doing is a self-supervised learning architecture. So the variational autoencoder that we use, that architecture does not need a lot of labeled images. It's able to identify from very few labeled images what is out of distribution. So it is able to tell you if there is a bird in an image when you're showing it a skin, or it'll just tell you like this is outside the data set if it's completely off the thing. So with that self-supervised architecture, if that is primarily used initially as this triage thing, and then you train a more specific model because it's identified like these set of images are totally out of distribution, then you train only a model specific to those ones. So for example, if you have a mammogram versus a chest X-ray, which are then more closer than say a bird image, right? So then you train a model on the mammograms. And this can also work on mammograms with certain kinds of calcifications versus lesions, right? They have been separated out as being of different classes. And then you can apply as a human radiologist labels like calcification or lesion on the mammogram, and then it gets trained on that. So it's self-supervised in that it's able to identify differences and then cluster them together that these are similar images. Now give whatever label you want to give it
All right.
in a more focused model.
Yeah, all right. I think I'll have to look into that paper also in more details, and that will give me more idea on how that works. And thank you so much for your excellent lecture. And due to the paucity of time, I would have probably taken a few more things. I think I'll connect with you, and I will get back. And thank you so much for being here. So I think, yeah. So I think our next speaker is also joined us. So Dr. Yan, can you hear me?
Yes.
Hi, Dr. Yan.
Yeah. Hello. Can you hear me?
Yes, I can.
Okay.
Okay, so please give me a moment to introduce you, and then we will start with your session. Okay?
Okay.
Okay. So moving on, we have the next session with Dr. I would like to invite Dr. Jingwen Yang, Associate Professor, Bioinformatics, Director, Bioinformatics Program, IOPUI, for our next session. Dr. Yan was awarded a CISE Research Initiation Initiative, CRII grant by the National Science Foundation for her work in developing a novel data-intensive computational framework to analyze and integrate data and networks, create predictive models, and reveal their associations. Her research has been on exploring brain imaging genetics associations by developing efficient and scalable computational and bioinformatics approaches. Specifically, Dr. Yan is interested in the combination of machine learning and network science such that rich biological knowledge can be properly incorporated to guide the learning procedure and help yield more interpretable results. We are delighted to have you with us today, and I now request Dr. Yan to kindly start the session.
Okay. Thank you for the nice introduction. So can all of you see my screen?
Yes, it is visible.
Okay. 1 second. Okay. So thanks for the nice introduction. So actually for today, so I'm doing like both brain imaging genetic association and multi-omic integration. So for today's presentation, I will mostly focus on one of our recent work on this multi-omic integration project. So it's called the Deep Transomic Network Fusion for Marker Discovery of Alzheimer's Disease. So for those of you who are not familiar with this disease, so Alzheimer's disease is one of the most common type of brain dementia. So there is many types of brain dementia like this Parkinson and Huntington, but the Alzheimer's disease is the most common form of dementia, accounting for around 60 to 80% of those cases. So for Alzheimer's disease, it's the bad part of that it's non-reversible and progressive, that it's going to slowly destroy the memory and thinking skills, and the patient usually will lose their capability of self-care ultimately. So there is two hallmarks of Alzheimer's disease in the postmodern brains. That means what the phenotype in the postmodern brain. So one is called the amyloid plaques. So basically, if you look at this figure, so these are the neurons from these normal aging adults or the healthy people. And on the right side are for the Alzheimer's patient. So you can see for Alzheimer's patient, these brown deposition between neurons are basically known as amyloid plaques. And inside the neuron, there is another hallmark called the neurofibrillary tangles. So these are two important hallmark of AD. And from imaging wise, like the Alzheimer patient has a much more severe brain atrophy. That means they have much smaller brains with very large ventricle in the middle. So these Alzheimer's disease is highly heritable, like based on the twin studies. So the heritability of Alzheimer's disease can be up to 80%. So there is a lot of effort trying to identify the genetic factors underlying the Alzheimer's disease. So these studies are known as genome-wide association study. And so there is a lot of them going on, and many genetic risk factors have been identified to be associated with a Alzheimer's disease. And these top SNPs are from the genes like ApoE, TOM40, and CLU. However, these genetic findings, these genetic variations, how do they exert effect to the downstream development of Alzheimer's disease is usually unknown, because they are mostly individual markers. They are not functionally connected. So we will not be able to know what's happening, like if a patient carries those variations, why they will develop a disease. So that means, taken together, we are facing the challenge to interpret and validate these genetic findings, and we will not be able to translate them to the targetable mechanism for disease therapy. So that's why it's kind of still their value in the clinical use are not so clear at this point. So on the other hand, we have a lot of multi-omic data in addition to this genetic variation. So we have a lot of multi-omic data collected for the Alzheimer's disease research like these. So these are all the possible data we can collect, but so far, like those large cohort, mostly collect data like these genetic variations, gene expression, protein expression, this metabolite, and also some of their demographic information, and these the social data, but it's very rare. So, and also in addition to that, we also have a lot of rich domain knowledge. That means we know which gene interact with another gene, which SNP is associated with which gene interacts with each protein, and which protein interact with each other, and as well as how a protein is involved in the synthesis of metabolites. So we know a lot of these prior knowledge, but so in our work, our goal is to integrate those multi-omic data together with those biological networks to discover the functionally connected multi-omic markers in AD. So why I emphasize on the functionally connected, because if you run a differential expression analysis, so you certainly is going to get these multi-omic biomarkers as well. But if they're not connected, so they are not functionally connected, so you still don't know why that happens. So you have no idea about the underlying molecular mechanism. So in this study, we use this data from this ROS map, which they have these genotype data, gene expression, protein expression, and also many other data points. We also we use the ROS map for discovery, and we also replicated our findings using an external data set called the Mount Sinai Brain Bank. So basically for both of these two cohorts, so the data, all the multi-omic data are collected from the same brain region called the prefrontal cortex, which is basically the front end of our brain. So these are the total number of proteins, genes, and SNPs, which are basically the single nucleotide polymorphism, a type of genetic variation. So these are total number of these proteins, genes, and SNPs. So why we only have these small number of proteins, genes, and SNPs It's because we have a small sample size. So we actually perform is a targeted analysis instead of proteome-wide or transcriptome-wide analysis. So now we look at all the data we have. Okay, so we have the genotype data, we have the gene expression data, we have protein expression data, and also we have this biological network as a prior knowledge. So inside that, we have for SNP and genes, we identify the SNPs that are located in or near the promoter region of each gene, which is 5K base pair upstream of, or say upstream of the gene. And for the gene protein relationship, that mostly comes from the reactome network, which is basically a protein interaction database. So when you look at all of this data we included, so you can see the highest dimension comes from the genotype data, like we're going to have tens of millions, which we will not be able to model them in the machine learning models. So we will have to do some pre-filtering so that we can fit all of this data into our model with that small sample size. So when you further look at this data, you see for the ROS map, there is a bottleneck in the protein expression part. Like they only measured around 186 peptides, corresponding to around 126 proteins. But for the genotype and gene expression, they covered everything. So in this case, we choose to use the peptide as seed, and then we identify the genes that are associated with those peptides. Okay. And also in the reactome, we identify the genes that interact with this peptide. Okay, so in that case, we get like two sets of genes in the gene level. For all of these genes, we identify the SNPs located on the upstream of these genes. So, and then finally, you see this is all the data we are going to include. Okay, so all these SNPs, genes, and peptides together with their relationships. So we come to our model once our data is ready. So our model, we call that this MOFNet. It's called this multi-omic fusion neural network. So we designed this neural network based on the central dogma of molecular biology that this information comes from, so the DNA is transcribed into RNA and then further translated into protein. So this neural network specifically incorporate this information flow from the DNA to RNA and protein, which you can see, which is in the first two layers of this neural network. So the first two layers, the first layer with the input as the SNP and genes and output as the genes. So in this case, so the genes are connected to genes itself. The SNPs are connected to the genes if it's located on the upstream of that gene. So in the first transparent layer, it tries to learn the information flow from the DNA to RNA, okay, which is the gene nodes over here. And similarly for the second transparent layer, so these connections are also based on whether this gene encodes for a protein that interacts with this protein. So these links are all from this prior network. So in this layer, we learn this information flow from the genes to the proteins. And then at the top, we have this fully connected layer for the classification. So for the first two layers, we use this L1 regularization because, so say like many of these links was going to get a weight. So the L1 regularization is going to enforce many of these links to get weight of 0 so that it's going to be removed out. Oh, we have talked about the, oh, sorry. So as I have mentioned, these links are exactly, oh, what happened? So the links in the first two transparent layers are basically exactly from this prior network we have curated. So that means once we learn this weight for each of these links, we will be able to map these weights back to this prior network and do a network pruning. So we benchmark our method with several other methods like this random forest and other logistic regression-based model using different penalty terms like the modularity constraint lasso, graph net constraint lasso, elastic net, and lasso. So these are the methods like can perform both prediction and feature selection. So that's basically why we select this. So all of those go through the same process for these parameter tuning, 5-fold cross-validation, same partition of subjects across methods, same portion of Hauser-Hammer and control participants across different folds. So first, we look at this classification performance of different kind of methods. So the first three methods we show here, the first three are all the MOFNet. As I have mentioned, the first two transparent layers are structured or wired using the prior network. So we examined three different kind of MOFNet. One is the MOFNet with curated network. And then we shuffle those prior network, but we keep those self connections to see whether the performance will be affected. And ultimately, we shuffle this network and also remove those self connections to see how much information we can get by purely integrating information from neighbors, but just completely disregard this information we collected. So overall, we can see like this MOFNet proposed get the best performance over all the other logistic regression-based method and also the random forest across accuracy, F1 score, this precision AUC, as well as specificity. But when you look at this MOFNet itself, so you can see once we shuffle the network, but keep the self-connection, the performance kind of is slightly worse than the one with curated network. But if we use the MOFNet with shuffled prior network and remove the self-connections, the performance becomes really bad. Like say, if you look at the F1 score, it's almost to 0 over here, and also for the precision. So in this case, we kind of get the conclusion that the collected multi-omic data itself does bear the most of the predictive power, but there's kind of some noisy, or maybe because of this snapshot of this expression data, we missed a lot of some information that we could possibly get from these neighbors, the functional interactors. So to identify which multi-omic feature we have selected. So we probe this prior transomic network using the weight we have learned. So this is the kind of the threshold we have selected with 0.003. So in total, we get 3 network components. And top risk gene ApoE, we also identified that, but it's not included in any of these network components because we did not actually identify a lot of functional interactions for this gene in the prior network. So this is the final 3 network components we have identified. So you can see these are basic 3 network modules. So one thing that is most important in our findings is about this transomic path. If you look at these nodes, these yellow nodes, there's the SNPs. So the green ones are the genes and the blue ones are the proteins. So if you look at these ones, you can see like, okay, so there are several passes like from these SNPs to genes to proteins, and also remember like, so this SNP connect to this gene, the link between them indicate that this SNP is located upstream of this gene and has a significant effect on the transcription factor binding activity. So that's our prior knowledge. That's why when you look at these transomic paths, So it's easier for us to interpret these findings that why it happens. So we look at many of these transformic passes. So we can say like, oh, there might be some cascade effect of genetic variations on the downstream genes and further regulate the protein expression. So we also further examine all the SNPs we identify to see whether they're going to indeed associated with the gene expression. So we did this expression quantitative trait loci analysis to identify whether the SNPs is significantly associated with gene expression. So in total, that 3 network components, we have 53 SNPs. And out of those, 3 of them were not found. Okay, but all the rest of 50, they are eQTOs in the frontal cortex tissue, which is where we get that all these expression data. And then we further look at these SNP gene pairs. So say these SNP and gene pairs in this component. So are they indeed? So say these SNPs, there are eQTOs, but are they the eQTOs of their connecting genes? So we examine all these 53 SNP gene pairs in the connected component, and we found like 29 of them are actually, those SNPs are indeed the eQTOs of their connecting genes in the frontal cortex tissue, which we highlight, we highlight in this figure with these blue edges, blue links over here. So we further did the pathway enrichment analysis to see what exactly are those modules. So we did that with this enrichment analysis with the pathway database Reactome. And these are the top pathways that we identified. One second, let me make this. So we find like these top rich pathways are the signaling notch 1, and there are signaling pathway, and inside that we see the significant ones, these which basically are the dark orange ones. So these are the most significant ones. One is this MHC mediated antigen processing presentation, and also here this proteasome degradation. This is a pathway that helps to clear these dead cells and misfolded proteins. So if you still remember in the first slides, like we mentioned, like one of the hallmark of Alzheimer's disease is amyloid plaques and tau tangles. So both of them are the misfolded proteins, and this pathway is responsible to remove them. Okay, so that's the part like we think is very interesting, that actually is going to connect in our dots to make a story more complete. And on the other side, we also find another pathway, that's top pathway, are related to this neurotransmitters release cycle. So inside this, so the most significant one are related to the GABA synthesis release and neurotransmitter release cycle over here. Okay. Sorry. So, and since our expression data are actually from this bulk tissue, so we also want to know which cells are those genes, identify the genes and the proteins. Where are they? Okay, are they specifically expressed in which cell type? So because each cell type usually has this marker genes. That means that those genes have selectively high expression in certain specific cells, but not the others. So we use that information to infer, or we say there's cell type enrichment analysis to say from these genes and markers we have marker genes we have identified. So which cell types are mostly related to those genes? So we did that, and we identified that those genes we have identified are mostly marker genes of astrocyte and microglial cell. And all of these two cells are these key cells in this central nervous system and support this function of neurons. And the neuron loss is one of these main functions that has been altered in Alzheimer's disease. So when we take a further closer look at all these network components we have identified, we found this component 3, which is exactly the core of this complex protein complex known as SNARE. So this complex is a key mediator of synaptic vesicle fusion. So if you look at this figure at the bottom over here, so you can see this is a membrane. So if you so the synapses are basically the synaptic vesicle fusion. So this is a vesicle that's going to translate these yellow dots. So these yellow dots will be the neurotransmitters. So this synaptic, this vesicle is responsible to transport this neurotransmitter from into the surface and then release it out to other neurons. So, and this SNARE complex is responsible for this vesicle fusion. That means fuse this membrane with this membrane so that it can open up and release those neurotransmitters. So this vesicle fusion process is really important for this communication between neurons and glias. So two important cells inside the brain. So, and when we perform the replication analysis, so we were able to find most of these proteins and genes in that independent data set, which is in the Mount Sinai Brain Bank. So for this one, we get around 82% of overall test accuracy, which is comparable as what we have got from this ROS-map data. And for most of those findings we have obtained from this ROS-map cohort, we kind of be able to replicate half of them for genes and proteins, but not so successful with the SNPs, which is kind of as expected. So to conclude, we have proposed a new deep neural network to jointly analyze the genotype, gene expression, protein expression, together with the prior biological interactions. So it helps us to prune the biological network and identify these submolecular networks associated with this disease outcome. It is more computationally more efficient than our earlier proposed modularity constraint logistic model, and also gives better performance on this unbalanced data. But so these studies still have several limitations that we're still working on. It's like we don't have a large sample size for the multi-omic data. So we are currently working on a transfer learning project that can help us to look for a potential solution for this issue. So, and it's built on top of these prior biological interactions. So it's not designed for a new discovery of interactions. So, and also it returns these subnetworks as system level biomarkers, which is easier to interpret, but more difficult for the clinical application. So one possible solution for that is like we can use the summary measures like this eigen gene or eigen protein inside each subnetwork as this marker, or we can use the upstream SNPs as this genotype. So to help with this patient screening in the clinical trials. So I would like to thank my collaborators, graduate students, and the funding support we have got. And I think that's basically all. And in questions.
Thank you, Dr. Yan, for an excellent presentation. And I don't see any question here. but I'll ask one question from you. So, considering the inherent heterogeneity in data types, sources, and scales that are across various omics data sets, what do you suggest that how we can use integration strategies to avoid potential pitfalls of biases and artifacts in multi-omics data analysis?
So what kind of it depends on this on how you integrate them. Okay, so in our case, like, so we basically when we build this when we build this model, that's something like we kind of acknowledge the noises and this inaccuracy inside this multi-omic data. That's why we incorporate the prior knowledge, and also we model that, we call this network fusion. We try to model this information flow from the DNA to RNA to protein, because say if the gene in expression data is not so accurate or might be noisy, so like it can be complemented by these kind of upstream SNPs, because sometimes these SNPs is also going to change or influence this expression of those genes. So we use that kind of information from another layer to I would say to complement this information that we have data we have collected to make it more predictive or more disease relevant. And for other kind of approaches, so if you are doing that step by step, you examine one omics data, then the other one follow just in sequence. So that won't be a problem, like when you, so say like these heterogeneity problem, like they're of different scales. So those kind of approach won't be a problem. In our case, if we put all of them together and examine all at once, we need to do the normalization. And sometimes, like if you want to look at these the same type, say the protein expression data for different cohort, you also need to do the harmonization process, which is pretty difficult. That's why we usually have a small sample size for the multi-omic data.
All right. Thank you so much, Dr. Yan. It was a wonderful presentation. And due to the paucity of time, I think we'll move on to the next speaker. Thank you so much. We are so glad to have you with us. Okay, so our next speaker today is Dr. Xiaofan Fang. Can you hear me? Hi.
Yes.
Yeah.
I'm here.
All right. So just give me a couple of minutes for your introduction and then we'll start.
Sure.
Okay. So now I would like to humbly invite Dr. Xiaofan Fang, Professor and Department Chair, Computer and Information Science, IUPUI, for our next session. Dr. Fang pursued his PhD in computer science from University of Utah. His primary research interests are in data visualization, biomedical image analysis, visual analytics, and volume graphics. His recent research interests are focused more on biomedical image analysis, visual analytics, theory and information visualization algorithms. These include visual analytics of brain connectome, data 3D facial image analysis for medical diagnosis, healthcare data visualization, and spatiotemporal visualization algorithm. We are delighted to have you here with us today. And now I invite Dr. Fang to kindly start the session.
Right. Thank you very much. Can you see my slides?
Yes, I can.
Wonderful. Thank you. So I'm going to briefly talk about my recent research in brain image data visualization and visual analytics. I'm going to it's basically an overview of this field called visual analytics. And recently we sort of extended that into AI and machine learning as well. So my application base is still brain image, which is quite similar to the previous presentation. It's in fact coming from the same project. So the some background about the image data we are using. There are two types of image data I'm looking at. One is the MRI data, then there is DTI data, which is the diffusion tensor images. Those are the white matter essentially in your brain, and the MRI data that we are working on are primarily the gray matter in your brain. So you use this to generate a connected networks that we're trying to visualize both modalities, the MRI data and DTI data, as well as the network data. So that's the background. So I'm a computer scientist, so my work is mostly on algorithm development applied on biomedical applications. So the three type of algorithms we focus on for this type of data would be start with data visualization. You basically visualize the images as well as the network information, and then you try to embed some analysis components into your visualization. We call that visual analytics. Then finally, we're trying to use visualization to guide machine learning. We call it interactive machine learning. Okay. So what is data visualization? If you're not familiar with this term, it's a fairly mature area. It's about developing algorithms and software systems for transforming data or images to visual representation, in other words, pictures, in a way that can enhance understanding, revealing insights of your data, right? And so I'm just going to give you some examples of our visualization work, because some of the algorithms are fairly standard. It's already mature technology, right? So this system that we built, so what you are seeing is a combined MRI data and DTI data. DTI are the fibers, the white matters. So how to use essentially transparency to sort of see both layers in the brain. And then here are the DTI fiber data. The color sort of represent the direction of the fibers in your brain. And then just some different variations of both MRI and DTI, as well as the interactions with different region of interest in your brain. And here is just an example of embedding network connections in this visualization. So those curves network edges of this connected network. So it's just different ways to how do you visualize different components of the brain as well as the network. So one of the innovative algorithm we developed here is, so this is new. So essentially, how do you visualize the whole brain? Because brain is a volumetric object. You look at it, brain, you only see the outside. So how do you see the inside as well as the outside, and how do you see it in one picture? So we use something called panoramic view of spherical raycasting algorithm. So this is an algorithm we developed. So idea is very simple. You basically put a camera in the middle of the brain. Then you let the camera, it's a spherical camera. So the camera will shoot rays on all 360 degrees. Then you use those rays to capture the brain components and then record them into a spherical surface, right? And then you open up the sphere, just like a world map. The world map is like that, right? It's a sphere. And we open it up into one image like this. So that's the so-called spherical raycasting algorithm. So just going to show you some video images here. So this is a small tool we developed to visualize. So these are the MRI data. Okay, different region of interest of the brain using color-coded images. And then here's we try to put a camera now in the middle of the brain. You can see, so you can move the camera around, and then what you'll see is essentially the spherical raycasting algorithm. Open it up, so you can even put the camera outside the brain. You can see different components of the brain. So it's a very flexible, interactive tool. Allow you to see sort of spherical objects or volumetric objects, right? So that's what this tool is all about. Let me see, it's almost done. So that's just the video for this particular algorithm. Oops. Of course, brain is a volumetric object. When you look at it, most of the time, even if with the transparency, you still don't see the entire interior layers. So we to overcome that, we use something called a layered rendering. So you see multiple layers of the brain using this same method. So you can see here, this is the outer brain, and the second layer, third layer, fourth layer, and so on. So that is so-called layers. You can put the layers together in one image with different color schemes and even network edges. Okay, so the whole idea is how do you really present all the information about the brain in one image using different techniques here, or visualization techniques here. So that's the first part of what I do. Visualize the data as well as some information within the data. All right. The second part is trying to embed a little bit of analytics in this visualization process. We call that visual analytics. It's again a mature field. It's been there for maybe 10, 15 years. It's about the use of sophisticated visualizing design and processes to analyze data sets and help users identify patterns and trends. So essentially, it's an analysis method, except you analyze the data through your visual cue, right? Your visual senses instead of automatically doing it. So human is the one to see and draw conclusions from those visualizations. So the question obviously is, how do you really design your visualization to allow human to gain insight from your visual cues, right? So there are many ways of doing this. I'm just going to give you one example here to use visualization to do feature detection. Okay, so the visualization method we're using here is the so-called multi-graph visualization. So we essentially treat each brain network. A brain network is a network. Right? A network is what? It's a graph, right? A graph typically is represented in computer science as a so-called adjacency matrix. So it's a matrix that record all the graph connections, right? So like this. So if each graph like this is a network, the network can be represented by a graph, by a matrix. So in other words, each person, if it has a network in your brain, you represent the network of that person's brain as one matrix, a two-dimensional matrix, right? So if you have multiple people, so essentially, for example, if you do classification in medical field, you have many subjects, human subjects. Some are, one group, for example, are normal people, then a group is people with certain disease. You want, in order to classify them or learn what's the difference between these two groups, what you do is take one group, you stack all the subjects' brain networks together, or stack the adjacency matrix together. So when you stack all those matrices together, it's kind of form a volume like this. It's like a cube, like a cubical volume. And then in visualization, there's a way to do visualization of this kind of volume. We call it volume rendering. So apply volume rendering, you can show this group of networks, how they look in three-dimensional space. But if you show multiple groups, you can see the differences of the groups. And then that tells you what areas are the differentiating factors that you can use as your features when you do analysis. Okay, so that's the basic idea. So again, this is a tool we've developed. You have the average. So we have 3 groups. Okay, again, this is for Alzheimer's disease and an Alzheimer's disease data set. You have multiple groups. Let me look at the data, show you the data first. So the data is like this. So essentially 3 groups, a normal group, then there's a mild cognitive impaired group, which are people who have sort of bad memory. And then there's Alzheimer's disease, diagnosed Alzheimer's disease group, okay, called HC, MCI, and AD group. So each group have different characteristics, right? So we have to pick combine all the groups into a volume, then you visualize the volume in the middle panel here, and then you can look at the differences of this group. For example, in this particular case, when I visualize them in the same angle, I do see clear differences in certain areas of this visual panel here. You can see, if I see some differences in 3 areas, then I will identify those as potential features. Okay, so once I identify that as potential features, then you apply essentially a standard classification method, the PCA or whatever visualization analysis method you want to use, using machine learning or SVM. Is all. I mean, the key here is the feature identifications here, feature selection here. So we use that. We apply the SVM with PCA and did a 3-fold cross validation on this particular data. So you do see improvements over the automatic feature selection process. So the right column are the automatically selected features. We use that to do analysis. The middle column are the ones with visually selected features. You see the classification results are clearly improved. So this basically shows that visualization can help with some of the analysis tasks, such as feature selections. Okay. So that's the second component I want to discuss. The third one is how do you, so that's a more recent work, how do you apply visualization in AI, okay, or particularly interactive AI. So here we, for example, focus on machine learning. So we all know machine learning is powerful, but it's a black box, right? A lot of times you apply machine learning, create a machine learning model, you don't really know how things happened and why it happens that way, right? So there's a huge trend in AI to generate something called explainable AI or interpretable AI, things like that. So using visualization in interactive machine learning will be one effort in achieving that goal. So here what we are trying to do is trying to visualize machine learning models and processes. Okay, for you generate the machine learning, you have a model which is basically implicit function of high dimension, right? How do you visualize that in a way that people can sort of understand how the machine learning model behave in your domain, right? Second thing is the machine learning process, for example, deep learning. The process is very abstract, okay? It's like a black box, so you have no idea what's going on here. So can you apply some visualization there to show people how the process actually work? So that's the goal here. we need to better understand the machine learning black box. We want to apply interactive human knowledge into the machine learning model process and improve performance. Okay, so a couple of challenges here. One is how do you actually visualize the model and the process in a space where people can understand, right? Second is how do you provide application context during machine learning processes, right? How do you see what's the intermediate layer, for example, in your deep learning, okay? And then finally is of course how human can interact with your machine learning model, right? So there are many different ways how to visualize machine learning models. I'm going to just present you one simple ones we used recently using a 2D heat map of scattered plot. Scattered plot is a visualization method, right, to visualize multidimensional data. So essentially, it's a two-dimensional subspace of your multidimensional data set, and then you project the data to that two-dimensional subspace. In our case, because we need to explain the machine learning model, therefore the subspace should be a space where people are familiar with. Okay, then I'm not going to get into the details of the algorithm, how these are actually visualized, because there are a lot of interpolation techniques involved in this. But I'm going to just show you the result here. So we apply this method to, again, human Alzheimer's disease diagnosis applications. So we use human brain connected networks, and again, use SVM classifiers for 3 classes, the same 3 classes. And then we want we generate a machine learning model of those classification model. So the key here is I want to visualize those models in a space where I can understand, or human can understand. So here we selected a few simple dimensions, we call it visual dimensions. So age, education, BMI, MMSE is sort of human cognitive scores, right? Those kind of stuff. Then you can see that here are the scatter plot, one snapshot of scatter plot visualization in that machine learning model. And so essentially, you see different, 3 classes is to see 3 different colors that represents 3 classes and the boundaries between them. And so you also add new samples. So you also visualize the sample training samples here. The purpose of this application is to try to see whether we could use fewer training points to gain the same visualization classification results, right? So the idea is we will start with a small set of training samples and generate a model, machine learning model. Then I'm going to visualize that model. Then I will see where in that space, in those space, I can add new samples to improve the model. So where do I add? Well, I want to add in areas with no or very few training samples. We also want to add points in areas with non-smooth model boundaries. If the boundaries are very jagged or very irregular, then you know that that boundary needs to be improved because the data may be not enough. Therefore, you add new samples or training samples there. Okay, so that's the idea, to try to use fewer training samples to achieve the same accuracy of the model. So it is a quick result of 2 different training process. The orange one is the one we use interactive models. So people add human looking at the visualization, add samples in our machine learning process. The blue ones are the automatic ones. So essentially, we're just automatically randomly add points. So you can see the accuracy increases as you add more points, obviously. However, the interactive ones, in other words, you add points more intelligently based on visualization results, you can see it can achieve basically the peak accuracy very quickly, much quicker than the automatic methods. So in other words, in a lot of training process, you actually don't need that many training samples. You only need a selected subset of it. The question is, how do you select those subset? Well, we're saying, well, you can do that by looking at the model visualization. So you visualize the model, that will give you some guide how to add points, right? So that's a very simple example to show how visualizing can help with understanding, even improve the performance of a machine learning model. Our last example is how to, let's say, use visualization to understand the process of your machine learning, in particular in deep learning. Okay, so you can see deep learning. This is a very simple deep learning model. Most deep learning models have a lot more layers, but I just want to show this with a very simple example with, say, 4 layers like this. Each layer has different nodes in those layers. We don't really know why those nodes got selected, right? Why the process used those nodes. What does those mean? For example, at the end, you only have 3 or 2 nodes. Why do they exist in that process? So we want to associate with nodes with the actual context or application context. For example, in this case, the brain components or region of interest in the brain. So that's the process. Now, how do you actually compute? So we want to say, well, look at each node. We'll say, well, what does that node mean? What are the coefficients or importance factor of different brain region of interest are represented in that particular node? So in order to compute those coefficients, there's an algorithm we call it backpropagation algorithm. So I'm not going to get into the details there. So you apply those, then you can say, okay, fine. You run a deep learning model. You click on any of the intermediate node. It will tell you what does this node mean. So, for example, this node means well, it's a coefficient, it's a combination of different region of interest in your network. And then you know, okay, what are the important one? For example, the red one is the very high coefficient for that particular component. Now, the purpose of this is to let the biologist or neuroscientist to look at this, saying, hey, this makes sense because I know this particular region in literature that is indeed related to this particular problem of Alzheimer's disease, for example. Or some may say, oh, this doesn't make sense. Something is wrong there. Okay, so this allows people to apply some interactive editing abilities. So you can go in to interactively edit some of the notes, saying, this looks really important. I want to add more note weights on it. Or this is wrong. We should delete it, and things like that. So it gives the user some power to input human knowledge, because the training process not necessarily encompass all the human knowledge, right? Sometimes human have intuitions or my knowledge about this particular problem that could be added into the training process through this particular interactive process. Okay, so that's the idea. Yeah, that's pretty much all. It's just an overview of some of this work that we did in this particular area. This is an exciting area, including data visualization, visual analytics, and interactive AI. So I hope some people will get interested and get into this particular field. Okay, thank you very much. Any questions for me?
Participants, you can paste the questions in the chat box, if any. All right, so I have one question for you.
Sure.
So can you tell me, like, when I'm using PCA, then how does the selection of different centering methods in PCA, such as mean centering, scaling, and the influence of interpretation of principal components and the variance explained by them can be dealt with? If you can tell me which scenarios, like, one are we should approach and which is more suitable.
I don't quite get your question.
So,
You said PCA. How is PCA involved here?
as in, how do I select the centering methods in PCA? I mean, I use mean centering or scaling. So which one is better? And if I can use something else for the PCA plot?
The PCA plot, I don't know, because PCA is a fairly standard method, right? So what do you mean by PCA plot?
So I mean scaling, if I am doing the scaling of the data.
Oh, okay. I am not sure. I think you may want to visualize it in a space that you could understand it better, and then make decisions based on your visualization results. So a lot of this are based on human visual senses, right? So it's hard to know before you see it. So I think the key is how do you visualize to make decisions based on that. So my suggestion is try to visualize some component of this data when you do scaling, for example. Okay.
Sure. Thank you so much. Sure. And it was an excellent lecture to have an understanding of everything that you talked about. And we are so grateful to have you with us today at this time.
Thank you very much. Yeah.
Thank you so much.
I just had Professor Fang, thank you very much. Sure. My camera is not connecting, so anyways, I will just talk. I just wanted to know from you that you have stacked the data and you've created these matrices. So could you just a little bit more explain that, like stacking, is it giving you more data which is more accurate, or what was the reason?
So we're talking about here, right? So we're saying that you can see that we have multiple groups of data set, right? So we have 3 groups. Each group have multiple subjects. Each subject is one matrix, right? It's one slice essentially. Because the goal is to see what are the differences between these 3 groups. But each group, you don't want to see just one slice, because you want to also make sure that all the people in that group have a commonality, right? Yeah. They have a common trail of certain features. So that's why I want to see the entire group. I don't want to see one representative in that group. So the way we see the entire group is to essentially put the network matrices together. You stack them, right? So you create a cube. Now, if you look at the cube, each slice or each 2D slice here is one person. When you put them together, it's a whole group. So when you visualize those things, you want to make sure that the same group of people here, they have the same features. They look similar. So those are the features that you want to make sure you can use. And these are the second group. They also after you stack together, they look similar in those areas, but they're different from the other group. Okay, so something like that. So you look for commonality within group, and you look for differences across group. Okay, so that's important. So the commonality part requires you put those slices together in a group, but otherwise you can't really see is this a common feature for all the subjects in that group or not, right? That's the idea.
Yeah, thank you very much for explaining that.
Sure. Yeah. Any other question?
I don't see any other questions. I think Dr. Priyanka, I think we can conclude for the day.
Yes.
We are really grateful to Professor Fang and all the colleagues at IUPI
Thank
to
you.
make it convenient for interacting with our students in spite of your schedule.
So I thank all the three speakers for the session, and it was great to have all of you here, and I'm sure the students must have benefited a lot with these sessions. So, ma'am, can we go on to close the session?
Yes, I think we should close now.
Yeah. I request the IT host to kindly stop the recording and close the session.